Welcome, everyone. This is Mr. Olson. Today, we're going to be talking about mutations. So, last week, we talked about transcription, translation, the process of protein synthesis. Yesterday, we kind of introduced genetics. Today, we're going to be talking about genetic mutations, which is going to take into place transcription, translation, protein synthesis. My apologies. So, we're going to go through the three types first. So, the first type is substitution, where one base is substituted for another. It's not always harmful and does not shift our whole amino acid sequence. So if we look at our original DNA, we have AAT, GTG, and then CCG. This gets substituted or has a mutation that now gives us AGT, and everything else after that's still the same. What you should take notice is that when we have a substitution mutation, it is only affecting one base. So it is only affecting one particular codon. It does not shift anything either one way or the other. Okay, so we are good here. The next one is called insertion. So insertion is when a base is added. Can it be more than one base? Yes. Is it typically harmful? Yeah, it is. And the reason for this is it actually shifts the whole amino acid sequence one way. This is what we call a frame shift. So if we originally had, wrong button. Timeout. We typically have, say, these three bases, and then we're supposed to have these three bases after, so this codon and this codon. When we insert things, that's no longer the point, or that's no longer the case. We are shifting that whole codon sequence over to one way, which is actually going to change our amino acid sequence, giving us a different protein, which typically is harmful. So this is insertion. We are inserting a new base. The next one is the opposite of insertion. This is deletion, and we are actually removing a base. This is also typically harmful because, once again, we're doing a shift. We're doing a frame shift model. So if we get rid of 3, this condenses everything. Or we move things, we condense. That means those amino acids that were originally supposed to be sequenced for, no longer going to get sequenced for, and we're actually going to make a different DNA. So we have substitution, insertion, and we have deletion. Now, we can do these 3 with just a couple easy things. Do substitution. All you need to do is count. Okay? We have, say, 9 codon or 9 bases right here in substitution. We're going to have 9 bases at the end. You have 9 and 9. You know it's substitution. Should you still find where the substitution took place? Yes, but this is the first step. Figure out which type it is. If you have the same number of bases in the beginning as you do in the end, it has to be substitution. Insertion, as you probably could guess, it's going to have more bases. So if we start off with 10 bases and we end up with, say, 12 bases, we went from 10 to 12, we added 2 more. That means we had to insert more bases into our sequence. And on the flip side, we have deletion. And this means if we start off with 10 and all of a sudden now we have 8 bases, this means we deleted some. So we have less of them. So just count. Okay, that's what we'll start off with. Now, how do mutations occur? This is just a rundown of substitution, insertion, and deletion. We're good to go here. This is just another visual for you guys to have. And it's actually showing some of the amino acid sequences down here, but we're going to talk about that a little bit later as well. So, mutations can have no or little effect, or negative effect, or a positive effect on an organism. And the reason for this is that our codon chart does have multiple things that actually code for the same exact amino acid. So, if we have CUU, CUC, CUA, CUG, all those codons realistically make the same amino acid. They make LEU. Now, if we had a mutation, originally we had CUC and now we have CUA, guess what? Our body's not going to notice it. It's not going to matter. But if we originally had CUC and also now it is CCC, now we're making this rather than this, and this is where we're going to have some issues. Okay? So when we figure out if it's going to have a problem or not a problem, make sure you look at the amino acids. So make sure you translate it first and see if it's actually changing the amino acid sequence. If it is changing the amino acid sequence, there's going to be an issue. If it's not changing the amino acid sequence, you're probably fine. Okay? Now, when we talk about protein folding, we understand that we have this amino acid strand, but how in the heck does it actually fold on top of itself is dealing with the words hydrophobic and hydrophilic. So your hydrophobic ends are going to attract each other, and your hydrophilic ends are going to repel each other. So in this image right here, we have this line, and then it's given us A, B, and C. Which one of these 3 is actually the proper folding of this amino acid chain? Your answer should be B. The hydrophobic ends attract each other, the hydrophilic ends repel each other. This gives us our 3D image. This is our protein. Now we're going to go through a couple examples of just what it might be, if what kind of mutations we have, just so you guys can see some actual visual examples. So our original sequence is CAT, GAT, CCA. This means we have 9 total base pairs. Like I told you before, let's just count it up right away. In our mutated sequence number 1, we have 3, 6, 9. So first and foremost, I can automatically tell you that this is a substitution mutation. Now, should I identify where it happens? Of course. So we have C, we have C, we have A, T, T, we have G, 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 A. So I know there was a mistake or there's a mutation on this A, which now became a G. Okay, is that going to affect our amino acid? Potentially. Do I have a codon chart in front of me? No, but it is something we could look at. Now, mutated sequence number 2 has 3, 6, 9. Oh, there's a 10th one. So if we have a 10th one, I automatically know that this has to be insertion. Now, should I see where this insertion took place? Yup. Okay, so let's do it. We have C, C, we have A, we have A, then also we have a T. But then I have a T over here. Okay, so this A right here must have been inserted, hence why we all shifted to the right. We had a frame shift. So it's going to affect all of our amino acids after, all of our codons after. And the last one is mutated sequence number 3. We have 3, 6, 8. So I know if I counted correctly, we have deletion. And where does that deletion actually take place? We had CAT, we had GGT, which is actually a substitution. Let me fix this. That is my fault. This should be GAT. Perfect. And then we had CC, and we got rid of the A. Okay? So, once again, there is a mistake there. That is deletion. Okay? So, what I want you guys to take a look at here, just so we're all on the same page, is that there are multiple mRNA sequences that code for different amino acids. So when you have a mutation, it is not necessarily always bad because you switch your mRNA sequence. Same or different mRNA sequences can code for different amino acids. Now, when it doesn't code for the same amino acid, that's where we start having issues and our proteins don't fold correctly, and then they don't have the right function, and there's not the proper thing. Okay. Now, there is some good mutations out there. One of the most famous one, and I'm not an expert in this by no means, is this evolutionary change in humans, and where the hemoglobin mutation named HBS that makes red blood cells takes on a curved shape or a sickle-like shape. With one copy of this gene, it confers resistance to malaria, so you're resistant to malaria, but with 2 copies of it, it causes the illness of sickle cell anemia. Now, having 1 copy of it is a benefit. Having 2 copies of it's not a benefit, but having 1 copy of it is a benefit, so it is a good mutation in that case. Okay, and those are some things that can be actually genetically passed on. That was a mutation. Guys, that is everything for today. What you need to make sure you take into consideration, or make sure that you guys have learned, is what is insertion, deletion, and substitution. If you're able to do those 3 things and be able to count, you got today. If you have any questions, please reach out to your teachers. Please reach out to me. Other than that, guys, have a good day. I will talk to you guys later. And bird next to pause. And adios.